the bunches should rest on the petiole of that of the plant, and while selecting the nut also we have certain criteria, like it should not be that much oblong, and it should almost be almost a round shaped one. And we have also discussed about the um, uh, the why we are getting coconut less than the net less than the potential level of a coconut palm. We are just very much below that yield potential of in India is very much below that of the individual palm is very much below. Why we have discussed about that. Then and we also have different colors of nuts and variations in the colors, and that and all we have discussed.
Anything else?
Then we have also mentioned that the female palm, that is spicata, then mainly that is are monoecious only, but the only specified that typically sir said about the spicata. Then
You can switch on, all of you can switch on your cameras if there is no issue. Yeah. The only thing is we feel more lively if there is a camera. Otherwise, as if we are listening to some radio or something like that. We don't feel that, though it's virtual, there should be some live elements in the class, right? We are not speaking to a blackboard. Yeah, that's better. Okay, Misbah, can you tell me why we have to go for embryo and pollen preservation in coconut? What is the significance of this cryopreservation of either embryo or pollen in comparison with the field gene bank or exchanging the seed nuts between the member countries of COGEN? What are the limitations and what are the advantages of these two methods?
If we preserve pollen or embryo, we can preserve it for a long time, long period, sir. It is advantage for us. We can preserve it for 10 years, 20 years, up to some extent we can preserve it. It is the advantage for us, but we can't preserve nut for long time, sir.
Why we can't preserve nut for a longer time? No, you can preserve the nut using cryopreservatives, like liquid nitrogen. We are using it for either embryo or pollen. We can preserve the nut itself in a cryopreservative. Is it not sensible?
But it is easy to preserve embryo compared to nuts.
No, because of the space which occupies. Is it not? It's very huge. Yeah. Then any other advantage of preservation through liquid nitrogen for pollen and embryo? So other than extending the life for a longer period, what are the other advantages?
Viability of the pollen.
No, when you are preserving means the aim is to regenerate plant out of either the pollen or the embryo at any later stage. It means, that's what it means, right? Without losing the viability. That's the basic objective of preservation, right?
Yes.
So other than this long shelf life, what are the other advantages of storing pollen and embryos. So I also mentioned about exchange of planting material between the member countries of Quadrant. So it's very easy movement, transport. You can send cryo vials to any other country, right? The lakhs of pollen grains you can send across, or you can receive also from elsewhere if it is a very genetically superior material, right? And all the time you cannot have you cannot maintain the germplasm in the field gene bank. It occupies very large area, and it requires a lot of financial requirements, and so as the labor force, is it not?
Yes, yes, sir.
Yeah, so it is less costlier, so you can extend the shelf life of either the pollen or embryo for a very, very long time, and for easy exchange of genotypes or the planting material between the member countries of Quadrant. So these are the major ones. Okay, and for later use, preserving embryo or the pollen in cryopreservation, you can use anytime you want in future. Because suppose if you keep pollen ambient conditions, the viability of the pollen grain is not even a week's time. That means you have to use it in your breeding or crop improvement programs. Suppose if you want to use it for, say, after 10 years, a genuine male parent, the pollen of a genuine, a superior performing male parent at a very later stage, you can conveniently use it out of this cryopreserved pollen grain. Is it not?
Yes, sir.
Is it clear?
Yes, sir.
Yeah, so there were many methods. One of the methods was, as mentioned here, and another advantage is the pollen and embryo are almost free from diseases. When you are storing a seed, there is probability of it may carry some pest or some disease, right? That risk will not be there if you are storing either pollen grain or the embryo. Correct. That's another advantage. Any disadvantage you can think of this cryopreservation? Any of you can answer.
If in between some microorganism, any
No, that's very. You mean contamination?
Contamination.
In vitro contamination. There is a probability if your hygiene protocol is not as per the standard, so there could be some kind of contamination. True. Any other disadvantage?
Maybe to bring like when we place a embryo for cryopreservation, then we are actually we are just actually giving a stopping a pausing for that embryo's normal metabolic activities. So
Correct.
when we again bring it back to normal, I think it will take a little time for adaptation.
Yeah, true. When you are preserving something at -196, you're almost stopping all metabolic activities in the embryo or in the pollen grain. So when you take it out and bring back to the normal ambient conditions, we may not achieve 100% results in terms of fruitful germination or further growth of the embryo and the pollen grain. So there is some, of course, maybe 10 to 15% of the embryos may not germinate later, or the pollen grains may not be viable after a long storage. Barring that, and barring what Joseph has just now told, there could be some in vitro contamination due to some microorganisms. And another issue is cost. So yeah, this cryopreservation compared to the, of course, in comparison to the overall preservation of the germplasm in the field, it is less only, but still it is a costly affair. Okay, so those are the disadvantages you can think of this cryopreservation. So you are extracting the embryo as per the protocol developed by CPCRI, and then you are desiccating, that means drying. Embryos are placed in a glass bottle which is filled with silica gel. Silica gel is basically which absorbs moisture from the atmosphere, right? So it dries up the embryos quickly, because any moisture present in the embryo or the pollen without proper drying, if you store, it will be subjected for microbial contamination during storage. Therefore, it has to be dried for a particular percentage. Okay, and lesser the moisture, more will be the shelf life. You cannot 100% eliminate moisture. That may kill the embryo or the pollen viability. That's all. So you should limit it to a certain extent. Then you have to incubate in the liquid nitrogen using cryo vials, which are around 2.5 ml volume. So you can use these cryo vials for preservation. And after putting them in cryo vessels, you can place them in a cryo sleeve and plunged rapidly into the liquid nitrogen container. Okay, and then whenever you want, so you want to take them back. So what we do is thawing. Thawing is the procedure when you take back from the cryopreservative. First thing you do is you will be keeping them in a water bath 40 degree for 2 minutes. Then before cryopreservation and after cryopreservation also, you have to do viability tests for both the embryo and the pollen. For pollen, generally the TTC is used, triphenyl tetrazolium chloride, and for pollen you can use astocornin. If the pollen or the embryo takes the dye or the color, it is indicative of viability of embryo and pollen. If it is not taking the dye, that means it's not viable. You can reject such embryos or pollens. So then they'll do in vitro recovery. Okay, of course, this was mainly contributed by Dr. Anita Karan, who is presently the director of CPCRI. Okay, because you just assume how easily you can store thousands of genotypes in the form of a pollen grain. On one single slide, if you place pollen grains, they are millions in numbers. So such is the requirement for their storage, such low space, ultra low space, what you can call. So that's why it's more advantageous. Suppose you just assume you want to plant 1,000 genotypes in the main field. How many acres of land is required in a field gene bank? You take 10 meter by 10 meter spacing. So in 1 hectare, you can accommodate 100 plants at that particular spacing. For conserving 1,000 genotypes, you need at least 10 hectare of area. It's quite huge. Here is just a small place, so you can easily preserve these genotypes for later use. Okay, because germplasm maintenance is a thankless job but very important job, so which occupies significant amount of place. This is in steps involved in pollen cryopreservation. So I already mentioned. So you are extracting the male flowers, drying them, separating and collecting the pollen grains, and then you have to assess the viability. Then you go for cryopreservation, and post-preservation, again you will test the viability or fertility of this cryopreserved pollen grains. And then you can use the viable pollen for further studies or crop improvement programs. Okay, so this is how they work out the pollen germination percentage. The number of germinated pollen grains, you can see the germinated pollen grains on the right hand side in the top right side picture with the total number of pollen grains. Okay, so multiplied by 100 will give you the percent pollen germination. So this is in relation to West Coastal and COD. They collected the pollen of these two varieties in March to May period for 3 years, desiccated to a moisture level of 7.5%. That is the limit standardized. And they are cryopreserved by direct immersion in liquid nitrogen. So germination and pollen tube length of desiccated and cryopreserved pollen were not significantly different. Okay, for both of these two parents. And then when they did pollen germination studies, it ranged from 24 to 32% in desiccated pollen, whereas it is 26 to 29% in cryopreserved. So not much difference. Okay. In case of West Coast all, germination ranged from 30 to 31. So those are the observations you can see. So normal seed set was observed in cryopreserved when they used cryopreserved pollen grain. That is what expected. Okay. So it should be viable, should be fertile, and it should be used like a normal pollen grain in the regular breeding programs. What we call this kind of storage facility as cryo banks. Okay, cryo banks. These advanced things may be asked in any of your exams, be it SRF or be it in ARS. So these are the ones you should be knowing, because other things you have already learned in your undergraduate courses. This exchange program is also important. I already mentioned there is the largest coconut germplasm exchange situated in Karnataka in a place called Kidu near Subramania on the way to Dharmasthala. Okay. Coconut seeds are highly recalcitrant. What do you mean by recalcitrant? What do you understand by the term recalcitrant seed?
Which can be stored only for 2 months.
Yeah, very limited period. Yeah. So you cannot extend the storage life of the seed for a longer time. So that's what recalcitrant, or rather they lose their viability quickly. They are called recalcitrant, and there is no dormancy. Okay. Like I think, can you say a few other crops where you have studied dormancy, or which plants or which seeds have seed dormancy?
Lupin seeds.
Lupins, yeah. Any other? Among the spices, important spice crops, which have tuber dormancy or rhizome dormancy. Do you think you can use the freshly harvested ginger or turmeric for sowing purpose?
No, sir.
Do you
No,
think
sir.
it will germinate?
No, sir.
No, sir.
Yeah. Most of the rhizomatous crops and also corms, bulbs, they have dormancy, right? You have to keep them for a particular period. Even if you plant them, they will not germinate until the dormancy gets over naturally. Okay, such thing is not there in case of coconut. That means they will readily germinate. It is both advantageous and disadvantageous. Okay, so these characters, especially the recalcitrant nature and lack of seed dormancy, they drastically limit the amount of material which can be gathered during collection missions. So the germplasm exchange is more difficult due to high risk of introduction of that's what I was just telling, why it is safer to exchange the pollen or the embryo which are devoid of any alien pest or disease. When you are, because most of the deadly diseases we have uh, got through these introductions only of planting material. When you're introducing a seed, when you're introducing a bulb or any other plant material, so if your quarantine is weak or not as per the expected standard, so you can get lot many diseases. So that's a big problem. To avoid that risk, we are going for the exchange of the preserved pollen or the embryos. So the embryo culture technique is also standardized, how to extract the embryos. For that, they'll select matured nuts of 10 to 11 months old. Okay. The collected embryos, first they are placed in distilled water and then they have to be sterilized. So they are using 50% chlorine water for 20 minutes for surface sterilization. After post-sterilization, again, they have to be washed thoroughly 4 to 5 times. There should not be any residues of this chlorine. And then they inoculate individually into the bottles containing 2 to 5 ml sterile water. So that's in detail. You can take later. So cryopreservation coconut embryonic shoot. Embryonic shoot can also be cryopreserved, like the pollen grain. Okay. Okay. Of course, we'll go back further. Yesterday, I told around 60 nuts is the national productivity. 64 to be more precise, as per the records. So I also mentioned certain allied coconut palms, the yield potential up to 300. So there are few very allied material which have the yield potential to produce more than 400 nuts also. So with the focus more on productivity enhancement, therefore we can only extend or we can increase the profitability. What the government is talking about, doubling the farmers' income. Okay, it's a very Herculean task. Definitely you can double the productivity, but we cannot say we can double the profitability. But when at the present market price, if you double the productivity, definitely the returns will be doubled. That's more logical rather. Suppose if you are getting say 100 nuts from one plant, if you could able, because of these technological interventions, if you could able to get say 200 nuts, so you are getting the almost 100% more yield. So that's very important. Excuse me. Can see the variability, different varieties, very high as far as stalls are concerned. Of course, in dwarf also, because of natural outcrossing to the extent of 20%, you can find a lot of natural variability, both in talls and dwarfs. Yesterday, I was mentioning about sweet kernel, aromatic water, and all those things. The one variety from this Mohachavon RL, from Ratnagiri region of Maharashtra, they were found to produce fruits with sweet endosperm. Okay, very important. This is what I was just referring. Pink fleshed coconut. So because of the anthocyanin pigmentation, this is a novel type. Of course, it was introduced. It was identified among one of the talls, which is called San Roman. It's an exotic collection. Now it is maintained at CPCRI Germ Plaza. You can see the pollen basal portion of the male flowers. You can also see the perianth region, and when you cut the pinkish coloration of the mesocarp is quite attractive. Of course, I'm not sure about its superiority in terms of mineral nutrients or the TSS. So, but nevertheless, people will be getting attracted to these colors. Okay, and other than that, attracting consumers towards the tender nut. So other applications you can think of is you can extract the natural color from the mesocarp. Of course, once they use the tender nut, so it will be considered as waste. So instead of wasting or instead of using it as a fuel load, you can commercially think of extracting the fibers from the mesocarp, which is naturally colored for making ropes and other things, right? Coir, colored coir you can extract rather, or you can extract the pigments if it is economically feasible. So this is called changarik, or just a second. You can see the biochemical estimation in normal and the pink colored coconuts. The first two lines for pink husked and the second two lines for the normal husked. You can see the phenolics in the pink husked phenols are slightly higher compared to the normally husked coconut. Phenolics, okay, that is one. Flavonoids are also on higher side, okay, 172 milligram per gram, whereas this is 145 milligrams in the normal husk. Anthocyanin, see, very important, very significant difference is with respect to the pigmentation. Anthocyanins in normally husked coconuts, the anthocyanin content is as low as 2.5 milligram per gram. So versus almost 14 milligram per gram in case of pink husk types. So that's why I said it's a novel type. So these novel types or echo types, what you can call, so can be characterized in situ and they can be preserved. You may preserve in terms of field gene bank, you can preserve their pollen, you can preserve their embryos in cryopreservation for a long-term use and use in your breeding programs. Okay. Okay, these are, of course, this is not a breeding course, but definitely an ideal palm. What you consider is definitely terms of superior performance with respect to yield. So we need to identify and we need to use better performing ones as far as yield of coconut is concerned. And wherever the copra making is a major commercial activity, for those regions, you have to have varieties or hybrids with higher copra and ultimately higher oil content. What is the normal oil content in coconut from the copra? Suppose if you squeeze or if you extract 1 kg of copra, how much oil you can expect? These are important things you should know practically. Okay. I'll give you 1 kg copra. How much oil you can give me? And how the oil is extracted? You have studied coconut, right, Sarah? Suppose you give 100 or 100 kg coconuts to a mill. So suppose if he gives, say 400, 40 kg of oil, do you accept?
No, sir.
Why you will not accept? You don't know what is the oil
Potential
recovery.
yield is of
No. I'm not asking about.
65%.
Yeah. Oil. Yes.
65%.
At least 60%.
Yes, sir.
60 to 65% is the normal oil
Oil
recovery in case of coconut. So you should invariably give, if you have given 100 kg copra, you should get at least 60 kg coconut oil. Will you come back or will you ask for some more, any other thing?
That cake, sir. Leftover cake.
The leftover cake is another important product. Okay. So generally they used to keep the cake and give the oil back and they are not taking any money in olden days. But now things have been changed. So now some charge is being fixed. Or if you are a coconut grower, a significant coconut grower, you can have small oil mills in your home itself. Now the concept is producing good quality oil at home scale. You may produce virgin coconut oil or you may extract the normal coconut oil. Why I am saying, what is the price of coconut oil? Any idea? Any idea? Coconut oil price, wholesale price. Why I am saying is, suppose if you sell the whole coconut as such, so maybe at the rate of say 20 rupees per nut. That's all. Suppose if you make copra on your own, say the copra is 150 grams and the present price is 17,000 rupees per quintal or 170 rupees per kg. So you work out, you will get copra, you will get mesocarp, you will get shell. All these things are additional products. So it is always sensible to convert it into a end product and then sell so that your net returns will be more like that. Okay. And an idea, EDO type. Have you heard the term EDO type? By any chance, have you heard the term EDO type? Not idiot type, EDO type.
Yes, sir.
What is the meaning? What is the meaning of an EDO type?
Specific.
No, it's like a model. It's like an ideal palm, right? Idiotype means ideal palm. So what is an ideal palm? Palm which is giving you very high yield, very high productivity, say more than 200 nuts per palm per year, more than 150 grams of copra per nut, more than 65% oil. So all these things are important, and the variety which produces flowering and fruiting earlier, like dwarf varieties or like the hybrids I have mentioned in yesterday's writing as well. And also we want a coconut variety or a hybrid which is resistant or immune to certain biotic and abiotic stress. So I said coconut will not tolerate moisture stress for a longer time. So as a breeder, as a scientist, you can identify a very good coconut variety or genotype which performs very well even under moisture stress. Drought tolerance. So how do you identify drought tolerant lines in any of the crops? What are the characters you look for in a drought tolerant crop?
Deep root system, sir.
Yeah, very good. That is one of the characters. So wider and deeper roots. Then
Small
Leaf
leaves.
area, sir. Smaller
Leaf area.
leaves.
Surface of the leaf.
okay, if leaf area is small, for photosynthetic efficiency will be less. It cannot be highly productive. Of course, what you said is correct. Okay.
Yes, sir.
Any other leaf characters?
Waxy coating or something on the leaves, sir.
Yes. Presence of epicuticular wax on the leaf surface. More the wax, less will be the transpiration. So the chances of the palm tolerating the moisture stress will be high, right? Okay. Any other characters for drought tolerance? Any amino acid in relation to drought? Have you heard about proline?
Yes, sir.
What is the role of proline?
It provides drought resistant.
Exactly. So these are some of the traits as a scientist, as a breeder, you look for in an idiotype to develop a variety which is tolerant to the moisture stress or drought tolerance. It's a very important area of research in future. Okay. Now, in many places, people are not growing coconut because of want of water. Okay, we don't have only all irrigated areas for cook. Even in irrigated areas, many times there is an issue of water. So to address those issues, as scientists, as breeders, so it's our responsibility to identify a very good variety or develop a very good hybrid. So which is having this particular drought tolerance. Among the pests, you see mites is a major one, major issue. Black-headed caterpillar is a major issue. Now, which is the pest of recent origin which is affecting the coconut? It is reported only during 2016 in Pollachi, in Kerala. Pollachi, Tamil Nadu, I think. Yes. Tamil Nadu or Kerala, Pollachi?
Tamil Nadu.
Yeah, Tamil Nadu. So it was reported from that particular area. Now it is widespread. The pest is widespread. Which is the pest? Very, very common now. Everywhere, that's the problem. It's a sucking pest. Rugose. Have you heard this term, rugose?
Yes, sir. White fly.
Rugose whitefly. Hope you have seen this pest. Have you seen?
Yes, sir.
You try to observe coconut palms beneath the leaves. Okay, you can
Yes,
find
sir.
this.
Whitish.
Yeah. So that's called rugose whitefly. So it's a serious pest of late. Okay. Like that, we need to develop varieties which can tolerate major pests and diseases. Among the diseases, of course, in Kerala and parts of Tamil Nadu, root wilt is major. In Karnataka, Ganoderma and stem bleeding is a big issue. Okay. Then varieties for different management conditions or cropping systems. You cannot have a same variety for all kinds of cropping system. The variety for a high density multi-species cropping system is different. A variety for monocrop or pure crop of coconut is different. Variety for moisture stress condition is different. Variety for different soil is different. Variety for different altitudes is different. So that's what region specific or location specific variety one should ideally look for. And triad specific lines, the last one. I just mentioned this pink fleshed ones. So, or aromatic nut water, or sweetish endosperm, or monoluron rich coconut, or Nira. You cannot expect all varieties to produce same amount of the Nira. Okay. Therefore, we need to identify superior lines. These are the characters of an ideal coconut palm or an idiotype. Okay. Idiotype concept is there for in most of the crops, essentially having important triads which I have mentioned just now. Can add some other triads also. Any doubts about this? Is it clear? Is it clear?
Yes, sir.
Okay. So constraints, these are some of the constraints in perennial crops and more so in case of coconut. What is limiting us in developing a real idiotype, a very good model or an excellent variety with all these traits? The main problem is the prolonged interval between planting and bearing, or the gestation period. So it's 6 to 7 years in case of talls, 3 to 4 years in case of dwarfs. So it is a restriction. You need to wait for so many times. Suppose you develop a F1 hybrid, you will identify the plants, mother parents, male and female. Then you will do crossing. Then you will wait for 10 to 11 months to harvest the nut. Then again, after harvesting the nut, you have to raise the nursery. You have to wait for one more year. And after that, you will plant the seedling again in the main field. So if it is a tall one, again, you have to wait for 6 years. See, at least for one generation, 8 to 10 years is the waiting time. Suppose if your service is 30 years, it's extremely difficult to develop only one or two varieties in crops like coconut. Okay, that's how. So we should reduce this cycle, the breeding cycle rather, by some means. The floral biology is distinct. So every month it produces new leaves, new flowers. So the nuts of varying maturity you can find on a palm anytime. It's highly heterozygous, highly variable because of natural cross pollination. Lot of variability. It is both positive and negative. It is positive because you can have scope for selection out of this natural variability. And then in case of dwarfs, not so highly variable because of more of self-pollination. But the extent of outcrossing is up to 20-25% in dwarfs also, depending on the wind. So that also you can employ. Then low rate of sexual, that's what I said. If you plant 1,000 nuts, you may get only the 500 good seedlings, which fulfills the criteria of a good seedling. Okay. Another big issue is, see, you compare cashew and coconut. Why in cashew we have very good hybrids, very good planting material? Why not in case of coconut? Because of this particular reason. That is lack of reproducible asexual methods. So there is no vegetative method standardized for coconut. That means you have to invariably propagate only using the seed nuts. So what is the advantage if it can be vegetatively propagated with respect to hybrids?
True to types.
True to type, and every time you need not produce the hybrid. Once you produce a hybrid, you can fix this hybridity through vegetative propagation, right? So you can keep on producing the hybrid seedlings without resorting to the hybrid seed production every time. You got this point? It's very important point. What I'm telling you is, you got this?
Yes,
So you
sir.
see in cashew, you develop a hybrid, then you can usually fix this hybrid through vegetative propagation, softwood grafting. So you can collect the plant science and you can do only grafting. So you need not do crossing every time and wait for so many days. So that is a very big limitation in case of coconut. Of course, now they have developed a protocol for tissue culture, but still it has to be commercialized. So it's a big problem in coconut. Hope it is clear for you. In mango, there is a vegetative propagation. Once you develop a hybrid out of sexual reproduction, you can easily fix the hybridity through softwood grafting or other methods of vegetative propagation. So the priority of you, maybe some of you wanted to become a scientist, you kindly look into this aspect in coconut. If you can develop a reproducible protocol for producing planting material vegetatively, that will be the biggest contribution and very challenging issue as well. Okay. Have you seen any other methods of propagation in coconut other than using the seed nut? Have you heard or have you seen?
No,
No,
sir.
sir.
Yeah. Very rarely it produces bulbils. You know bulbil?
Yes, sir.
What is a bulbil?
The fleshy
Underground.
Yeah, not bulbs, bulbils. Bulb are different, bulbil is different. Have you seen the plant agave?
Yes.
Yes, sir.
How many times it will flowers in its life cycle?
one.
Yeah, once it is flowered means it is going to die. Before the death of the agave plant, it produces quite huge number of bulbils, which are actually vegetative structures, which will produce the new plants, right? Similarly, this concept was reported in coconut, but unfortunately, you cannot use it to our advantage. The results were not so encouraging, and it is not a reproducible character in all the varieties. So we have to think of only tissue culture. Okay, and long period of experimentation required to obtain the results. It requires more land, more labors. So it's a cumbersome. So these are some of the constraints and improvement. So these are the different kinds of varieties you can have: local varieties or cultivars. Okay, improved varieties for released by institutes. Then of course hybrids. You have different combinations. You can have D into T. You can have D into D. You can have T into D. You can have T into T. Why we have to have all sorts of combinations? Can you say something about this? Why we have to cross a tall with a dwarf or vice versa? Why you want to cross?
To get good hybrids.
What is a good hybrid? What is the problem with tall?
Cross
No, what is the major constraints in tall varieties?
harvesting.
So harvesting is a big issue in crops like coconut and arecanut. So if you want to reduce the palm height, then you want to cross T with D or D with T to reduce the height.
D with the T, sir.
Why not the other way? What will happen if you cross a tall with a dwarf?
The plant will occupy the characters of its parental T, sir.
You mean to say this height is contributed by the maternal parent?
Yes, sir.
So therefore you have to go for D into T, right? To reduce the height. Okay.
Yes.
So why, how to improve productivity of talls by using dwarfs? The problem with tall is it takes more time for producing flowers and fruits, right?
Yes, sir.
The gestation period is more. How do you reduce the gestation period? By employing these dwarfs. The advantage of dwarf is it is precocious. Right. Just 1 second. Hold on. Hello. Yeah. Namaste, madam. Yeah. Save mark. Illa, madam. Illa. Now already I'm having one big loan. I cannot afford any other loans. Yeah. Thank you. Yeah. Now you tell me. How to address the problems of the talls and problems of the dwarfs. Problems of the tall, as you said, one of the problem is its height. So you can develop hybrids which are semi-tall, which are dwarf. Using dwarf as a female parent is one of the concepts. That's clear, I suppose. Jyotsna, is it clear?
Yes, sir.
Okay. Now tell me, what is the other problem with the talls? Compared to dwarf, the productivity of talls is less. Dwarf will produce more number of nuts per palm per year in contrast to the talls. How can you improve this particular trait using dwarfs?
Pollen of dwarf plant is crossed with tall gynoeciums.
It was a T into D, you mean to say. You're crossing a tall with a dwarf, taking tall as a female parent. So you mean to say the high yielding character of the dwarf to some extent will come to the next generation if you do so, right? Yeah, that's one of the strategy. Yes, you can improve the productivity of talls by using dwarfs in a breeding program. That's why T into D hybrids. Second one, right? So D into T, why we are crossing D with the T is reduce the height.
Height of the plant.
T into D to improve the productivity. Why you want to cross a tall with another tall? Why do you want to cross? Okay, I'll give you some clues. Say, for example, this Arsikera or Tiptur tall. Say, imagine the oil content is only 60%. So there is one more variety, local variety, which is a tall variety called Lacadive ordinary, LO. The oil content is more than 70%. Does it make sense to cross these two to improve the oil content in case of popular tall varieties of coconut? Does it make sense? I want to improve the oil content in, say, the regular East Coast tall, West Coast tall, or our Arsikere and Tiptur talls. How can you do that? It's a qualitative trait, right? Unlike yield, oil content is a qualitative trait. It is possible. So, and they have developed several hybrids with higher oil content by crossing a tall variety with another tall variety after identifying a suitable pollen parent and female parent. So that's one of the strategy. So why you want to cross a dwarf with another dwarf?
Quality.
So disease resistance or precocity or any desirable traits if you want to combine. Okay, I think you heard general combining ability and specific combining ability that will be studied before selecting the parents. Is it clear why we have to have different kinds of hybrids in coconut?
Yes, sir.
Maybe now the concept of this NERA tapping is being highlighted of late. So we may have to evolve varieties which can give you more NERA yield with better quality parameters, more sugar content and all those things. Okay. Another kind of natural variants in coconut, as we already discussed, were NCDs, natural cross dwarfs. I told the name of Dr. Patel in Kerala. Neeleshwar is the station where the first coconut hybrid was developed. That was based on his observation. he has raised the nurseries of the dwarf. He could able to see around 20% variation in the nursery, and the variation was due to natural outcrossing. So the same concept has been utilized for developing the hybrids in coconut. And of course, we have exotic varieties also, like San Ramon tall, Philippines ordinary, all those things. So any other kinds of varieties you can think of other than what is mentioned here? Local types, improved ones, hybrids, NCDs, exotic cultivars, any other? No GM so far in coconut, unlike rubber. Is it clear?
Yes, sir.
Any doubts so far? Any doubts? Sir, on 10 minutes, ಮುಗಿಸ್ಕೊಡ್ತೀನಿ. Yes. So, how well you participate, how well you interact, how well you pose questions will lead to some good learning. It's not only listening. As student, you should be very curious to know, to dig deep based on your previous understanding. Okay. Okay. So many hybrids and varieties have been developed already. The list is very exhaustive. See here, dwarf into tall hybrids. Here, dwarfs are taken as female parents and talls as male parents. The distinct advantage of this D into T over T into D is that could be produced on a large scale by regularly emasculating the dwarf mother palms, permitting free natural crossing. We are not doing crossing. We are just emasculating the dwarf forms and allowing natural pollination between dwarf and tall in an isolated place. Otherwise, this manual crossing is very, very laborious. If you are planting only two varieties in isolation and emasculating only the dwarfs and allowing for natural pollination, still it is a hybrid. If you properly do emasculation process, D into T hybrid. That's one way of reducing the cost in the hybrid seed production. But big challenge is having the isolated area. Otherwise, there is a lot of pollen contamination. You do not have control over the pollen parent if you are not isolating. Okay, that's how it can be easily produced. Then use of orange or yellow as female parent is one of the important strategies, as it's easy to identify the hybrids based on the petiole color in the seedlings. Otherwise, you have to use biotechnology tools for confirmation at a nursery stage. You don't know whether you have done hybridization properly or not. Okay. Before evaluation, so all these things have to be done. Yellow, orange, and red petiole colors are recessive traits in comparison to the brown and green pigments. So in hybrid seed production. Okay, why T into D we have already told. Here talls are taken as female parents and dwarfs as male parents. So generally, COD and Gangabondam, they are considered as best for producing economic hybrids with West Coast tall. That combination is superior. Okay. The advantage of this particular T into D, you see, they take only 4 to 5 years for bearing, unlike the regular tall cultivars, which take 6 to 7 years for first inflorescence production. One is precocity in bearing, and the second thing is increased yield compared to the regular talls. Okay, and copra quality is also good. See, around 176 gram and oil recovery of 70%. It's really superior. And the hybrid palms are easily susceptible to soil moisture fluctuation, resulting in button shedding and drooping of leaves during summer. So when you are choosing hybrids for planting, so invariably the most important requirement is they will not tolerate moisture stress like the regular tall counterparts. So don't ever suggest the coconut hybrids for a moisture limiting situation. Being scientists, you should be aware of these things. So yellow, that is Lacadive ordinary, is used as female parent. Hybrid exhibited drought tolerance and better yield. Why? Because Lacadive ordinary, highest oil producing one, and epicuticular wax content is very high. Root distribution is really wider and deeper compared to other tall varieties. So if you take that, the advantage is you can expect some kind of drought tolerance in the produced hybrids. T into D production is more time consuming and laborious because you have to do emasculation, hand pollination, and all those things. Okay, is it clear?
Yes, sir.
Okay. Of course, we heard about NCDs. Okay, they are open pollinated progenies of dwarf with the talls. Okay, D into T only. They are like natural D into T. So they are present to the extent of around 20%. And you can select the hybrid seedlings based on the petiole color as marker. Okay, and then they are highly vigorous. Why T into T? So this also we have already explained. So produced by intravarietal hybridization of tall with another tall. So individual palms having better breeding value are identified, and these genotypes are grown on isolated seed gardens used for the production of T into T hybrids. For combining distinct traits of talls, quality with yield, quality with drought tolerance, yield with drought tolerance, and many other traits. Though they are late to bear like regular talls, but the advantage is their yield potential is high. You can improve the yield potential of talls by producing T into T hybrid. That's a big advantage. Okay. So these are, I think we will stop here. We'll continue in the next class. Okay. Any doubts in today's class? You have any doubts? You are free to ask. Any doubts? Anyway, I have recorded the session. I may share if required, because it's better you make your own points based on the class discussion. Addition to that, you have to collect some authentic new information from the resources which I have already mentioned yesterday. So that you will have sufficient resources for the preparation for your exams, any exam, be it SRF or be it ARS or any other competitive exams, even for the semester exams. Any doubts? Misbah is worried only about oil palm. Sarah, I have given you some small assignment yesterday. Be you able to attempt? Sarah?
Hi, sir.
Yeah, you can just
So,
tell about
but
that.
we can. So we have the particular formula for the calculating the planting distance, which is dividing by the area into the spacing which has been recommended by. But the farmers usually adapt by they standardize. They use ropes or pegs to plant and maintain distance. They usually measure by feet. So for example, in our farm,
Okay.
my father has not only my father, so no one used to follow the spacing which have been recommended in books or in a scientific manner.
No, I
They
am
used
not
to
asking what is
go-
recommended. I am not asking what is recommended. What was the area under each crop in your farm, and how many plants they have planted? Hello? Hello, am I audible? So I could not hear anything. Hello. Hello, there was some connectivity issue. Now you can continue, sir. Yeah.
So we can calculate the number of plants or plant density by dividing the area into spacing. So for example, we can calculate for the hectares. For hectare, there would be 10,000 square meter. If we take the planting distance of 10 into 10 meter, we have to calculate with that, and then we will get about 100 plants per hectare.
No, I'm not asking the procedure. What was the situation in your farm?
So I'm not getting. So can
No. You said you have some arecanut plantation, some coffee and some rubber, right?
yes, sir.
I was just asking
sir.
what is the area
They are
under each.
planting in the quincun system.
Okay.
In between the 4 rubber plants, they have.
Yeah, tell me. Between 4 rubber plants.
So in between the 4 rubber plants, they have planted one areca in the middle one, sir. Like
You forget about arecanut. What is the spacing for rubber between rows and between plants?
They have maintained the wider spacing, so about 8 meters.
Okay, 8 by 8.
Yes, sir.
Have you measured?
No, sir. My father has told, sir.
Oh, you're not verified. Okay.
Sir, the farm is about 15 kilometers from my home, sir.
So you cannot
even
go
my
there. You don't
No,
want
sir,
to
I
go.
was unaware. Sir,
Okay.
not like that, sir. We have many function in our home, sir. The father is busy in that. So before coming to the college, I will go and
Better. Before
Yes, sir.
reporting here, go to your farm.
Yes, I will go and
Yeah, you physically measure yourself all the spacings
Yes, sir.
and total area and total plant population. Okay.
Yes, sir.
Yeah. Thank you. That's all for the day. If you have any questions, you are free to ask. Otherwise, we'll end. Any questions?
Sir, you told about rolling, no, sir?
Yeah.
I don't know, sir, what about that rolling?
No, the accumulation for this particular amino acid will be more in moisture stress. That is one of the indication that the plant can tolerate the moisture stress. If you measure its content in varieties which are drought tolerant, if you just compare tall and dwarf, it will be more in case of talls compared to dwarf. That's one of the indicators for drought resistance, and you can characterize the genotypes for its content. So whichever the genotype is having higher proline content, it is expected to have better drought tolerance. That's what it means. Okay. Yeah. Any other question? I think Misbah, you can collect more information on that particular aspect and share in the next class about proline and its importance in drought tolerance. Okay. Yeah. Jyotsna, any questions? No. Okay. Sarah?
No, sir.
Okay. Thank you. You can leave. Yeah.
Thank you, sir.
Yeah.